But there are some organisms, especially the green plants, which cannot directly utilize the atmospheric nitrogen or the nitrogen present in the soil. This nitrogenase enzyme is highly sensitive to oxygen molecules. They get degraded if it is subjected to oxygen molecules. The relationship between the bacteria and the legume plant is one of the best examples for the symbiotic relationship. Hello everyone, a warm welcome to the last session of this chapter, that is Mineral Nutrition. I am Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysore, Temple of Excellence. So in the previous session of this chapter, we had discussed about the nitrogen metabolism, wherein we understood about the nitrogen cycle and how plants fix atmospheric nitrogen by taking the help of various bacteria, that is the chemoautotrophic bacteria that are present in the soil. In this particular session, we shall in detail discuss about the biological nitrogen fixation. So to begin with, we shall first understand how this biological nitrogen fixation actually occurs. So biological, it means something living or a living organism is involved in the process of nitrogen fixation, therefore making available of nitrates to the plants. So very few living organisms are capable of making use of the atmospheric nitrogen directly. So whatever nitrogen is available in the air, that is abundantly it is present in the air because of industrial pollutions, because of the natural nitrogen that is present in the air and so on. So these nitrogen can directly be utilized by various organisms directly. But there are some organisms which cannot make use of the nitrogen directly. The best example is the green plants. So here, as I was saying, there are few organisms which can make use of nitrogen directly. They are some of the prokaryotic species. So these prokaryotic species are capable of fixing nitrogen, and there are certain prokaryotes which are capable of directly fixing or making use of the atmospheric nitrogen. But there are some organisms, especially the green plants, which cannot directly utilize the atmospheric nitrogen or the nitrogen present in the soil. They have to convert the nitrogen should combine with hydrogen to form ammonia. That ammonia has to combine with oxygen to form the nitrite, and the nitrite has again should combine with oxygen to form the nitrate. So that is how they get converted, and that nitrate is being utilized by them. So this particular process, if it has to occur, then the role of different living organisms are involved. So reduction of nitrogen to ammonia by living organisms is called biological nitrogen fixation. So what did we understand? Some of the organisms, especially few of the prokaryotic species, are capable of directly fixing the atmospheric nitrogen, but some eukaryotes such as the green plants, they are not capable of directly fixing the atmospheric nitrogen. Therefore, they need to depend on the other organisms to do so, and that is called as a biological nitrogen fixation. And this biological nitrogen fixation, if it has to occur, or if the ammonia or if the nitrogen has to get converted into ammonia in particular, then there is an enzyme that is required, and that particular enzyme is called as the nitrogenase. So this is one of the important components of the prokaryotes, because I told you there are some species of prokaryotes which can directly fix the or directly make use of the atmospheric nitrogen, wherein in their body itself they produce an enzyme called as the nitrogenase enzyme. This nitrogenase enzyme converts the nitrogen to ammonia, therefore making it available to them, and such microbes are called as the nitrogen fixers. So what are nitrogen fixers? Nitrogen fixers are the microbes that are capable of making use of a nitrogenase enzyme to convert atmospheric nitrogen to ammonia. So here I told you nitrogen, it is made up of two atoms which are held by a triple bond, and therefore it is a strong bond. So this bond has to be broken. They have to combine with hydrogen in the presence of a nitrogenase enzyme and form ammonia, that is NH3+. So this is how ammonia gets formed, and this is the process that is done by the nitrogen fixing bacteria or nitrogen fixers. So we shall understand about the different nitrogen fixers that are there in the environment. or in the soil. So the nitrogen fixing microbes, they can either be free living or they can be symbiotic. Free living means they do not have any association with the plant. Their only function is to convert the nitrogen to ammonia and then make it available to the plant. But symbiotic nitrogen fixers means they depend on the plant. The plant depends on these bacteria and the bacteria depends on the plant. So they are showing a symbiotic association. So both are benefited. Here it is called symbiotic bacteria. So some of the examples of free living nitrogen fixing aerobic microbes are the Azotobacter bigerunkia, and also one more, an anaerobic free living microbe is the rhodospirillum. So we have Azotobacter, which is aerobic nitrogen fixing bacteria. Then we have Bigerunkium, which is also aerobic nitrogen fixing bacteria. Then we have one anaerobic nitrogen fixing bacteria, which is rhodospirillum. So rhodospirillum is anaerobic in nature. That is, it doesn't require oxygen for its survival. And not just that, there are also certain cyanobacteria. So cyanobacteria are nothing but the blue-green algae, which also helps in fixing the atmospheric nitrogen. That is why these blue-green algae are added to the crop field, because they help in fixing atmospheric nitrogen, such as Enabena. We have Enabena here and nostoc. So these two are the blue-green algae, that is the cyanobacteria, which are free living and they help in fixing the atmospheric nitrogen. So next, let's talk about the symbiotic bacteria. So far we talked about the free living bacteria and the algae that helps in fixing the atmospheric nitrogen. So there were two types of aerobic bacteria and one type of the anaerobic bacteria. And also we understood how they are free living, because they do not form any symbiotic association with the plant. They just fix the nitrogen and that nitrogen will be utilized by the plant, but they don't derive any benefit from the plant. So here now they harm the plant. So here, talking about the symbiotic nitrogen fixation, the relationship between the leguminous plants, that is the beans, then we have various leguminous plants, especially the beans, peas, all these are the leguminous plants. So these leguminous plants, if you pull out the leguminous plants, in the root you can see knot-like structures or nodule-like structures. So you can see here these nodules that are formed. So in these nodules, the rhizobium bacteria tend to live. So the relationship between the bacteria and the legume plant is one of the best examples for the symbiotic relationship. So here the species of the rod-shaped rhizobium bacteria, as you can see, the rod-shaped rhizobium bacteria has a very good relationship with the roots of a majority of the leguminous plants, such as alfalfa, sweet clover, sweet pea, then lentils, garden pea, broad bean, clover beans, etc. All the bean varieties that are there. So all those have a good symbiotic association with the rod-shaped rhizobium bacteria, which helps in the formation of the nodules in the root, and not just that, they therefore helping in nitrogen fixation. So the most common association of the root is as root nodules. So these nodules are nothing but, as you can see, they are small outgrowth that you can see on the roots. And apart from that, so nodules are very, very important, which you find in only the leguminous plants. So that is why the rhizobiums, they help in nitrogen fixation in the leguminous plants. And there is one more microbe which is called as Frankia. So this microbe also helps in nitrogen fixing, production of the nitrogen fixing nodules, but they do not help in the formation of nodules in the leguminous plants. They help in the formation of nodules in the non-leguminous plants, example the alnus. So Frankia, these are also free living microbes and they also help in nitrogen fixation, forming nodules. But the only difference is they don't form nodules in the leguminous plants. In the non-leguminous plants, they help in the formation of nodule-like structures, as you can see. So this is Frankia, which is again a free living bacteria. So both rhizobium and Frankia are free living in soil, but as symbionts, they can fix atmospheric nitrogen. So they have a symbiotic association. So what these do, they derive nutrition for the plants, and in turn they convert atmospheric nitrogen to ammonia and then to nitrites and nitrates and give it to the plant. So that is how both are beneficial to each other. They form a symbiotic association. So how exactly this occurs, we shall see. So to understand this, first we should clearly understand what nodules are. So nodules are nothing but they are near spherical outgrowths on the roots, which are seen in both the leguminous plants, and also if frankia is involved in the nitrogen fixation, in non-leguminous plants also you can find this nodules. So if you cut through the nodule, so you can see small almost spherical like structures which are found in the roots of the leguminous plants, which are the nodules. So if you cut through the nodules, you can see a central portion which is pink in color. It is pink in color. So we know the color of hemoglobin, right? Hemoglobin is it provides the red color to the blood. That is, RBCs are red in color because of the hemoglobin. Just like that, these nodules, they contain a compound called as leg hemoglobin. So leg hemoglobin. So this leg hemoglobin is actually the one that gives this particular color. So here in the leguminous roots, that is nodules of the leguminous plant, when you cut them, you can notice that the central portion is red or pink in color. It is red or pink in color because of the presence of leguminous hemoglobin or the leg hemoglobin. So you find them in the nodules of the leguminous plants. So why are these important? We shall study later, but you have to know what is the nodule and what is present in the nodule. So that is why I had explained it here. So we understood that nodules are nothing but small, almost spherical kind of outgrowth on the roots of the plants, and when you cut the nodule at the center, you can see a red to pink color, and that red to pink color is nothing but because of the presence of the leguminous hemoglobin or the leg hemoglobin. And this leguminous hemoglobin, what function it has, we shall see later. So let's understand, because we know now what nodules are, we shall understand how exactly these nodules are formed. So nodules are formed by the rhizobium bacteria, right, and the frankia in the case of non-leguminous plants. So let's see the development of root nodules in the leguminous plants, that is in the soya bean. One example here, soya bean. So rhizobium bacteria, you can see here, where are the rhizobium bacteria present? In the soil particles. So in the soil particles, lots of rhizobium bacteria will be present. So these rhizobium bacteria have to reach the root hair. So if it has to enter into the root, first it has to reach the root hair. You have the epidermal region of the root, if you can see. From the epidermal region, some of the epidermal cells, this you'll easily understand if you have watched the anatomy of flowering plants. So what is epidermis, what is root hair, all that you will understand. So this root hair, some of the cells get converted into root hair. This root hair actually helps the roots to properly absorb the mineral nutrients, water, etc. So these root hairs, so the bacteria, what they will do, that are present in the soil surrounding the root, they will start moving towards and collecting towards the root hair. So once they start collecting towards the root hair will slightly curve or it will bend. So as it bends, or it will form a hook-like structure. As it forms a hook-like structure, the bacteria starts entering into the root hair, forming a thread-like infection structure and entering into the cells, and they will enter into the inner cortex and the pericycle cells, which are under division, and there they will help in the formation of the mature nodule. So now what is there in this nodule? Because of the division of the cells that are occurring and the entry of the bacteria and all that, the nodule or extra growth, that is nodule-like structure, will get formed on the root. So that is how the root nodules usually get formed. So what happens? The rhizobium bacteria comes in contact with the root hair and it will start to multiply there or divide there. As it divides, as a large number of rhizobia bacteria gets formed, it successfully infects the root hair, therefore causing the root hair to curl. And once the root hair curls, the infected thread will be formed along the root hair, carrying the bacteria into the inner cortex. So cortex, just below the epidermis, what is present in the root? It is the cortex, right, which is made up of parenchymatous cells. And the bacteria there gets modified into rod-shaped bacterioids. So that is why rhizobium bacteria are rod-shaped. Inside the root, once they enter the cortex, there they will get modified into rod-shaped bacteria, and they will cause the inner cortical cells, that is the cells of the cortex and the pericycle, to divide. So when they divide uncontrollably, what happens? The division and the growth of the cortical cells and the pericycle will lead to the formation of extra cells. These extra cells are nothing but which forms the nodule. Like how tumors and all are formed. They are extra growths, right? Because of uncontrolled division. Here also extra division of cells will take place in the pericycle as well as the cortex, leading to the formation of the nodule. So that is how nodule. So a mature nodule is complete. Why? It is complete because it has vascular tissues, that is the xylem and the phloem, and it is continuous with that of the root. So that is why. Because nitrogen has to also be carried, that is nitrate should also be carried. So they have to have a connectivity. That is why they also have a vascular tissue which is connected with the tissues of the root. So this is how the nodule formation occur. So we know the structure of the nodules now. We know about how the nodules got formed. So because we know all that, we shall further understand how exactly what happens in these nodules. If nitrogen fixation has to occur, what exactly happens in the nodules? So the nodules will contain all the different types of necessary biochemical compounds such as enzyme. Which enzyme? Remember, first I had told you nitrogenase. Why nitrogen is important? In the presence of nitrogenase enzyme, the nitrogen gets converted into ammonia. And the first step in the nitrogen fixation is conversion of nitrogen to ammonia, right? So here nitrogenase enzyme will be present in the nodule, and also in the nodule what will be present? Leg hemoglobin. Why leg hemoglobin is present? Because I told you when you cut the nodule, at the center of the nodule you can see a pinkish to reddish color, which is because of the presence of the leg hemoglobin. So what are each of these functions? We shall see. So the enzyme, that is nitrogenase, is nothing but a protein which is made up of a moiety, that is molybdenum iron protein. And this molybdenum iron protein, that is the enzyme. What is the function of an enzyme? To catalyze the rate of a reaction, to increase the rate of a reaction. Therefore, the nitrogenase enzyme, which is nothing but made up of a protein, that is molybdenum iron containing protein, it actually catalyzes the conversion of, or it increases the rate of conversion of the atmospheric nitrogen to ammonia. So the first stable product that is formed during the nitrogen fixation is ammonia. So the reaction which occurs is as follows. What happens here? Nitrogen combines with 8 molecules of electrons and therefore with 8 molecules of hydrogen in the presence of 16 molecules of adenosine triphosphate to form ammonia. If all this has to occur, what is important? Nitrogenase enzyme. Nitrogenase, therefore releasing water molecule, and here the dephosphorylation will occur. 16 ADPs will be released and therefore 16 inorganic phosphates will also be released. So this is how ammonia gets formed. So we can see here the steps of conversion of atmospheric nitrogen in the nodule, how it will occur, we can see. So here they have taken the enzyme. So this is the enzyme. What is the name of the enzyme? Nitrogenase. Why nitrogenase enzyme is required? Nitrogenase enzyme, where it is present? It is present in the nodule. So in the nodule, now the bacteria are acting. What is the bacteria is doing? It is making use of the nitrogenase enzyme and converting the nitrogen to ammonia. So the nitrogenase enzyme, where it is there? Inside the root nodule. So the nitrogenase enzyme in the root nodule. Now the plants, now what is there? The nitrogen. So nitrogen, from where it has come? Nitrogen gas, that is from the atmosphere in the form of water droplets and all. They would have reached the soil and the plants are now absorbed. So the nitrogen will enter into the nodule. So here, that is in the soil, the bacteria will start to act upon it. So the substrate is nitrogen gas. So nitrogen gas, I told you, it is made up of 2 nitrogen atoms which are held together by strong triple bond, right? So you can see here a triple bond, that is nitrogen having a triple bond. So this nitrogen will be taken in by the nitrogenase enzyme, and that nitrogenase enzyme, what is the function now? Can you see here the nitrogen is taken in by the nitrogenase enzyme. The nitrogen will go and bind to the nitrogenase enzyme. In the nodules, the nitrogenase enzyme will reduce the nitrogen into ammonia. As you can see here, it combines with two molecules of hydrogen to form NH2. So it has formed NH, a bond NH. So this is what it has formed. Now this ammonia that is there, again it will combine with two molecules of hydrogen to form NH2. So with a single bond NH2. So can you see how the bond getting broken? So very easily the rhizobium, by making use of the nitrogenase enzyme, it has helped in breaking the bond here. So NH2 will be formed, and again now NH3, ammonia is NH3, right? So again hydrogen molecules, two molecules of hydrogen. Why two molecules of hydrogen are added? Because for nitrogen is made up of two atoms, right? And for each of the atom, the hydrogen has to get added. So here two molecules of hydrogen. Therefore, if you can see here, what will happen? We have NH3 and NH3. So N with one H here, second, third. Again this atom with one, three of its hydrogen. Therefore, forming ammonia. Now what happens? The ammonia will get released into the soil. The ammonia will get released into the soil, and whereas again the nitrogenase enzyme, what is there? So from the nitrogenase enzyme, the ammonia gets released to the soil, and these nitrogenase enzyme, again what it will do in the nodule, if it is present, it will be released in, it will be left over in the nodule, and from there again it will combine. It will come here, again it will combine with the nitrogen. So therefore, this process will continue, the cycle will continue as such. So this is how exactly the nitrogenase enzyme is involved in the process of conversion of the nitrogen into ammonia, and that is to ammonia, wherein ammonia will be released. Well, next what happens? The ammonia gets converted into nitrite and then to nitrate. So we shall look into it. What is the next formation? So the enzyme, that is nitrogenase, is highly sensitive to molecular oxygen. So why nodules should be formed? Now you can ask me the question, without nodules all this could have occurred, right? Why was there need for the formation of nodules? So I told you, where is the nitrogenase enzyme present? In the root nodule. So this nitrogenase enzyme is highly sensitive to oxygen molecules. They get degraded if it is subjected to oxygen molecules. So therefore, it requires an anaerobic condition wherein there is no oxygen, and that so the nodules have such an adaptation that it ensures that the enzyme is protected from the oxygen. So that is why nitrogenase enzyme is present in the root nodules. So in the root nodules, nitrogenase enzyme is protected from the oxygen, and to protect these enzymes, the nodules will contain an oxygen scavenger called as a leg hemoglobin. Now we understood the connectivity, right? So what is a nodule? Nodule is a somewhat spherical like structure which at the center contains a pink or a red color pigment called as the leg hemoglobin. So the nitrogenase enzyme that is there inside the nodule is protected by the oxygen scavenger, that is leg hemoglobin, wherein whatever oxygen is there, it will be made use by the leg hemoglobin, therefore not letting the oxygen molecules go near the nitrogenase. Why? Nitrogenase cannot survive with the oxygen. They get degraded if oxygen is there. So that is why leg hemoglobin protects the nitrogenase enzyme from the oxygen, by that itself making use of the oxygen. So these microbes that are there, they live as aerobes under free living condition where nitrogenase is not operational. But during nitrogen fixing events, they become anaerobic. So these are aerobic bacteria. Rhizobium and all are aerobic bacteria. So these rhizobium, they need oxygen. So one half of the time they live in the oxygen condition, and when it's time for nitrogen fixation, they get converted or they evolve themselves into the anaerobic form. So that is how the nitrogenase is protected, and in the nodules they convert the nitrogen into ammonia and the further processes will occur. So this is about the nitrogenase enzyme and why leg hemoglobin is important. So next, let's understand what is the fate of ammonia. So now we understood that ammonia is produced in the root nodules. So what is the fate of the ammonia here? So the ammonia at physiological pH, the ammonia is protonated, that is it combines with the one hydrogen or a proton to form an ammonium ion, because whatever is absorbed by the roots of the plants are in the form of ion, calcium ions, molybdate ions, sodium ions, right? So here what happens, I told you, in the nodules, first the nitrogenase enzyme will be present. The nitrogenase enzyme will convert the nitrogen to ammonia, that is NH3. That NH3 will be released to the soil and the nitrogenase will remain in the root nodules, wherein it will again enter into the repetition of the process. Now the ammonia that is there in the soil, it has to be absorbed by the plants in the form of ions. So therefore in the soil at a particular pH, the ammonia gets protonated or a proton gets added to ammonia. Therefore, the NH3, when it gets protonated, it forms NH4 plus. It forms NH4 plus. So while most of the plants can assimilate nitrate as well as ammonia ions, the assimilation of nitrate is, that is the assimilation of ammonia ions is quite toxic to the plant. So that is why it has to get converted into nitrate and then be utilized by the plant. So what it means is most of the plants can assimilate nitrate as well as ammonia. But this ammonium ions which is there in the soil, if it is taken in by the plants, it might prove toxic to the plants. So that is why the ammonia has to again get converted into nitrates and then utilized by the plants. So we shall see what is the role of this amino ions or the ammonium ions in synthesizing amino acids, because I told you nitrogen is very important for the plants because it is one of the important constituents of the amino acid. Chains of amino acids are required for the formation of proteins, and proteins are very important for all the biological activities in any of the living organism, right? So here, what is the role of ammonium ion in synthesizing amino acids in plants? It can be done in two ways. One is reductive amination and the other one is transamination. So what did we understand so far? We understood that ammonia cannot directly be taken in by the plants because it may be toxic to the plants. So that is why the ammonium ions that are there has to be reduced or converted into nitrates and nitrites. And this ammonium ions are in turn used for the formation of various amino acids in plant. How does this occur? By two important steps, that is reductive amination and transamination. So we shall see what these two are in detail. First is reductive amination. So reductive amination means ammonium ion get reduced. So here in this process, the ammonia reacts with alpha ketoglutaric acid. So where is this alpha ketoglutaric acid? It is one of the intermediate component of the Krebs cycle. So Krebs cycle will be occurring inside the plant, and that Krebs cycle, one of the intermediate component is alpha ketoglutaric acid. So that alpha ketoglutaric acid will react with ammonia which is taken in by the plants and it forms glutamic acid. And this glutamic acid is then being utilized. So we'll see here by the equation here. So alpha ketoglutaric acid, which is a component of intermediate of the Krebs cycle, it combines with ammonium ions in the presence of NADPH and also in the presence of an enzyme glutamate dehydrogenase to form glutamate releasing water and NADP. So now glutamate is formed. This glutamate is what? This is the reductive amination. Ammonium ions got reduced to glutamate. So glutamate is got. Next is transamination, the conversion of the ammonia. So it involves the transfer of amino group from transamination means the change, the transfer. So here the transfer of amino group from one amino acid to the keto group of a keto acid. So what happens here? That is the transfer of amino group from one amino acid to a keto group of a keto acid. So here talking about glutamic acid. So glutamic acid is the main amino acid. From where did we get the glutamic acid? The ammonium ions that are absorbed by the plants is actually toxic to the plants. Therefore, the ammonium ion combines with alpha ketoglutarate acid to form glutamate. Now, or to form glutamic acid. So now this glutamic acid is one of the main amino acid from which the transfer of NH2, that is the amino group, takes place, and the other amino acids are formed through transamination. So you can see here we have the amino donor and the amino acceptor. So one of the amino group is accepting the amine, and whereas the other one is donating. So that is what actually occurs here. So the enzyme transaminase, it actually catalyzes such a reaction. So here what happens? The glutamic acid, which is a main amino acid from which the transfer of amine. So here you can see here, from here the transfer of amine occurs to a different group. So it is transferring, the amino groups are getting transferred here. So this is about transamination. So this is the fate of ammonia at the end, wherein reductive amination as well as the transamination will occur. This is important because the plants cannot keep ammonium ions for a longer time as it is toxic. It was about this particular session wherein we understood about the biological nitrogen fixation, wherein the rhizobium bacteria that is involved in the formation of the root nodules, and how in the root nodules the nitrogen gets fixed, especially that is the nitrogen gets converted into ammonia, and ammonia finally it cannot be good for the plant directly. Therefore, it gets converted into, it combines with alpha ketoglutarate acid in the presence of glutamate dehydrogenase enzyme forming glutamate or glutamic acid. This glutamic acid, which is one of the main amino acid, it actually helps in the transfer of the amino group to the other components. That is nothing but the transamination process. So this is how the biological nitrogen fixation occurs. So with this particular chapter comes to an end. I hope you have understood the whole chapter. We shall meet again with new sessions taking up a new chapter. Thank you.